Picture this: a forestry drone mapping a burn scar in northern Ontario flags something its algorithm can't classify. Not damage, not debris. 7 shapes, standing dead center in a fire zone that should have taken everything. This isn't a news story. It's a thought experiment. What would actually happen, scientifically, if a tree species was reduced to just 7 individuals on Earth? I built this scenario using real conservation science, real propagation techniques, and real disaster response protocols. Stay with me, because the science here is almost stranger than fiction. In our scenario, it starts with a drone pilot doing unglamorous work, mapping a burn scar in northern Ontario for an insurance survey, the kind of contract job that pays the bills between real projects. His name is Marcus. The canopy mapping software flags an anomaly. 7 shapes, clustered, unclassified. He almost ignores it. Sensor glitches happen constantly in scorched terrain, but he flies back out the next day anyway, more out of curiosity than anything, and finds 7 conifers standing in a ravine the fire never reached. Bark unlike anything in his field guide, deep braided ridges, needles growing in fused clusters instead of singly. He clips a small branch, bags it, and drives it 2 hours to the nearest university biology department. The professor who examines it won't commit to anything. Too little material, too strange a texture, nothing in the regional database to compare it to. She sends the sample out for genetic sequencing and tells him it'll be weeks before anyone can say anything for certain. 3 weeks later, the results come back, and the room goes quiet. It's not a damaged sample of a known species. It's something the genetic record says shouldn't exist anymore at all, a lineage last seen in the fossil record roughly 20,000 years ago, during the last glacial maximum. Word moves fast after that. A second team is sent to verify the find independently, this time collecting bark, needle clusters, and a small resin sample without disturbing the root systems. It takes another 4 months before a formal paper is ready. The researchers want to be certain before they put a name on something this significant. When the paper finally goes up, it doesn't just report a rediscovered species, it reports 7 of them, alive, in a country that's been ecologically surveyed for over a century, which raises the obvious, uncomfortable question: how does something like this stay hidden for so long? The answer turns out to be almost mundane. The ravine is steep-walled, shaded most of the day, and sits inside a wildfire evacuation zone that's kept hikers and surveyors away for years. Sometimes the rarest thing in the world survives simply because nobody had a reason to look. 7 trees, 1 ravine, 20,000 years of silence, broken by a drone doing routine insurance work. Here's the problem conservation biologists would immediately face in this scenario. 7 individuals isn't just rare. It's below the threshold most population geneticists consider viable at all. The real-world benchmark, the 50/500 rule used in conservation genetics, suggests a minimum of 50 individuals just to avoid immediate inbreeding collapse, and 500 to maintain long-term evolutionary adaptability. 7 is roughly 14% of even the short-term survival floor. So the first question isn't how do we protect them? It's, are they even genetically distinct from one another? Or are we looking at 7 clones of a single ancestor? Because if they're clones, one pathogen could end the species in a single season. In our scenario, the sequencing comes back mixed. 5 of the 7 share nearly identical genetic markers, suggesting they're root sprouts from a single parent tree rather than separate individuals in the strict genetic sense. But 2 trees are different, slightly different lineages, distinct enough that a geneticist on the project flags them immediately. That distinction matters more than almost anything else in this story. 2 genetic threads instead of 1 means there's a mathematical chance, however small, of introducing real variation back into the population instead of just multiplying a single genome. The team runs the numbers on what a rescue would actually require. To reach even the low end of genetic safety, they'd need to grow the population by a factor of 7 or 8 within a decade, using cutting propagation rather than seed, since collecting seed risks damaging trees this irreplaceable. There's also a quieter problem nobody says out loud in the first meeting. Even if propagation works perfectly, all the new trees will still trace back to the same two genetic threads. You can multiply survival odds. You can't manufacture genetic diversity that was never there to begin with. It's a strange kind of victory condition. Not save the species in the way people usually picture it, but buy the species enough time and enough copies that a future disaster doesn't erase it entirely. Almost immediately, the discovery creates a second crisis. How do you study something without accidentally destroying it? In our scenario, the first research visit nearly ends in disaster. A support helicopter, sent to lower equipment into the ravine, drifts too close on approach and clips a low branch on one of the 7 trees with its rotor wash. No one is hurt. The tree survives. But the incident report that follows changes everything about how the site is managed. From that point on, access becomes tightly restricted. No published coordinates, no geotagged photos. A short list of approved researchers, entering on foot only, using sterilized boots to avoid introducing outside pathogens or seeds. Even provincial wildlife officials are only told the general region, not the exact ravine. The reasoning is blunt. A species this fragile can survive fire, cold, and 20,000 years of isolation. What it might not survive is a single careless visit from the people trying to help it. This is the part of species rescue work that rarely makes headlines. Not the discovery, not the science, but the unglamorous discipline of simply staying out of the way. With the site secured, the real work begins. Figuring out whether this species can even be propagated at all. For most conifers, cuttings are treated with synthetic rooting hormones before planting. A decades-old standard practice. In our scenario, the first batch of treated cuttings produces inconsistent, disappointing results. Some rot. Some simply do nothing. A junior researcher on the team proposes testing the opposite approach. No hormone treatment at all, just controlled humidity, filtered light, and patience. It's considered a long shot, the kind of idea senior staff tolerate more than endorse. The untreated batch outperforms the treated one by a wide margin. Of 50 untreated cuttings, 34 take root within 10 weeks. The treated batch, by comparison, manages barely a third of that. Nobody fully understands why at first. The eventual working theory is that this species, having survived for millennia through a stress-adapted physiology, responds better to being left alone than to intervention, which turns out to be a fitting metaphor for the entire rescue effort. Within 8 months, the lab has 34 young clones growing under controlled conditions, genetically traceable back to the 2 distinct parent lineages identified earlier. For the first time in 20,000 years, this species exists outside a single ravine. It feels like the hardest part is over. It isn't. 18 months after the propagation program begins, a routine inspection turns up something alarming. Soil samples from the ravine test positive for a root rot pathogen, never previously recorded in that watershed. The likely source is human. An unauthorized hiker, spotted on a trail camera weeks earlier, despite the access restrictions, is suspected of having carried spores in on boots or gear. For a species already surviving on a mathematical knife's edge, a soil pathogen is close to a worst-case scenario. Root systems this old and this interconnected. Remember, several of the 7 trees may share root structures entirely, mean a single infection could spread underground before anyone notices symptoms above ground. The response is immediate and drastic. A chemical barrier trench dug around the healthy root zones. Soil testing expanded to a much wider radius, and for the first time, discussion of whether the wild population needs physical fencing, something the original secrecy-based strategy was specifically designed to avoid. 6 tense weeks of monitoring follow. The infection, it turns out, hadn't yet reached the root systems of any of the 7 original trees. Caught early, contained. But the scare accomplishes something important. It proves, for the first time in this story, that the ex situ population, the 34 clones growing in a lab 3 provinces away, isn't just a backup plan anymore. It's now, functionally, an insurance policy the wild population might one day depend on entirely. Then, 2 years into the program, the real test arrives. A lightning-sparked wildfire, the kind that's become almost routine in Canadian summers, starts 60 kilometers away and begins moving toward the ravine. Not aimed directly at it. Wildfires rarely move in straight lines, but close enough that fire behavior models give it a real probability of reaching the site within 4 days. This is the scenario every species rescue program eventually has to plan for and hope never happens. Aircraft drop retardant in a containment ring around the ravine's upper edge. Specialist crews rappel into terrain inaccessible by road to run irrigation lines, soaking the ground so that if fire does arrive, it burns as a slow, cool ground fire instead of a canopy-consuming one. Everyone involved is asked to keep the operation's exact location out of any public reporting, even after the fact. The secrecy protocol holding even under emergency conditions. The fire reaches the ravine on day 3. For nearly 36 hours, thick smoke makes aerial confirmation impossible. Ground crews can't get close enough to check without risking their own safety. Then, on the morning of day 5, smoke clears enough for a drone, flown fittingly by Marcus, the same pilot who found the grove in the first place, to get a clear pass over the ravine. 6 of the 7 original trees are still standing, scorched at the base but alive. 1 did not survive. It's a real loss. It's also, against every reasonable expectation for a population this small, a survival. The fire changes the program's long-term strategy. Protecting one ravine, no matter how carefully, means the entire wild lineage still depends on a single point of failure. In our scenario, researchers select a second site. Similar elevation, similar shade cover, deliberately chosen far enough away that no single wildfire could threaten both locations at once, and begin transplanting a portion of the lab-grown clones back into wild conditions. It doesn't go smoothly at first. Roughly a third of the transplants fail to establish in the first 2 years, mostly from transplant shock rather than any flaw in the cloning process. But the 2/3 that do survive begin, slowly, to behave like a genuine second wild population rather than a managed nursery planting. It's a strange milestone to celebrate. Trees planted deliberately by hand, in a location chosen by a spreadsheet rather than by 20,000 years of chance. But for a species that came this close to existing nowhere at all, it represents something close to a second chance at having a chance. Here's what's strange about writing a scenario like this. Almost nothing in it is invented from thin air. The genetic math, the propagation science, the pathogen risks, the fire response tactics, every mechanism in this story is drawn from real conservation programs working right now to save species that have been reduced to a handful of individuals. The only fictional parts are the location and the species itself, which raises a real question, not a speculative one. How many species like this are we not finding? Species this rare have been discovered before in places assumed to be thoroughly mapped, hiding in terrain simply inconvenient enough that nobody had reason to look closely. If it's happened before, in countries surveyed as extensively as ours have been, it's worth asking what else might still be out there. Not extinct, just quiet, waiting on the other side of a burn scar nobody thought to fly a drone over. If a story like this is the kind of thing you want more of, real conservation science, told through scenarios that make the stakes feel personal, that's exactly what this channel is going to keep doing. Subscribe and turn on notifications, because the next one might be about a species that's already out there, still waiting to be found.